I would like to welcome all attendees. I will present my research under title Mutation Effect of Chemical Mutagen Ethymthane sulfonate EMS on the Vitality of some Yeasts, submitted by Dr. Badia Abdul Razzak, Malla, Obaida, College of Science, Department of Biology, University of Mosul, Iraq. Introduction. Yeasts are microscopic eukaryotic organisms located within the fungi kingdom. Their dimensions range from 3 to 10 micrometers in diameter to 5 to 13 micrometers in length. It belongs to under 3 fungal subdivisions: Ascomycota, Basidiomycota, and Deuteromycota. 1,500 species have been described to date. It separates in many food and can have a major or secondary role in the spoilage of some foods. The latter case results from the fact that it grows at a relatively low rate compared to bacteria. Mutagens work to bring about a change in the arrangement of nitrogen space, whether by deletion, addition, or substitution, which is either substituting a base for another plus from the same group, and this is called a transition, such as replacing a purine base with another plus in the same group, or a base pyrimidine is another plus. But if a purine base is replaced by a pyrimidine base or vice versa, the process is called transversion, which leads to a change in the composition of the gene. And thus a change in the composition of the protein that encodes it, which may result in a change in characteristics such as the inability to synthesize some amino acids or vitamins, or the emergence of allergy or resistance to some antibiotic and others. Ethyl methyl sulfonate is one of the alkylating agents that attacks the CH3CH2 and methyl group at the 7th site on the purine ring. This removes the base by displacing the DNA without affecting the sugar phosphate column. Due to the importance of this product to the consumer and the seriousness of its contamination with these microorganisms and the financial losses it causes, the current study focused on isolating and diagnosing some types of yeast that contaminate it, and studying the effect of chemical mutagen ethyl methyl sulfonate on its vitality. Materials and methods. Isolation and diagnosis of yeasts. Collected 30 samples of orange juice from local market in Mosul City, Iraq. It was diagnosed based on the phenotypic and biochemical characteristics referred to earlier. Mutation effect of chemical mutagen ethyl methyl sulfonate EMS. Germansen 1983 was used to study the effect of the chemical mutagen ethyl methyl sulfonate on yeast viability. The number of surviving colony and the percentage kills were collected using the following equation: percent survivors, the resulting number from mutagenesis treatment, divided by the resulting number of non-mutagenic dishes multiplied 100% kill equals 100 minus percentage of surviving individuals. Antibiotic resistance test of wild type and mutant yeast isolate. For the process of determining the resistance of non-mutagenic and mutagenic yeast isolates using an ethyl sulfonate. The solidified yeast extract peptone agar medium was prepared with the addition of antibiotics as the final concentration mentioned in candizole, clotrimazole, fluconazole, ketoconazole, lamizole, and nystatin at a concentration of 30 micrograms per ml. The isolates were inoculated by streaking method and the plate were incubated at a temperature of 28 for 48 hours. Isolates are antibiotics as a mutant isolates that are resistant to the antibiotic use here can be identified. The result. The current study demonstrated the presence of 25 isolates of yeasts out of a total of 30 orange juice samples belonging to 4 different genera. The most frequent yeast was Rhodotella rubra, which appeared in 9 isolates by 36%, and the least appeared was 4 isolates in Trichosporon Asia with 16%. Figure 1 shows the characteristics and microscopics of yeasts. Rhodotella rubra, Trichosporon Asia, Cryptococcus laurent, Candida tropicalis. The results of Table 2 shows the ability of all yeast isolates to grow at temperature of 25 and 37, except for Cryptococcus laurent, which was growth negative at 37. The table data also revealed the ability of Rhodotella rubra and Cryptococcus laurent was shown to benefit from nitrate as the only source of nitrogen as it grew weakly, and the remaining 2 isolates were tested negative. Regarding the determination of the ability to resist acetic acid, all isolates were negative for the test, except for Rhodotella rubra, which was positive for the test. As for the test to determine the viability of growth in low level of water with a rise in the level of carbohydrates, it was found that Rhodotella rubra and Trichosporon Asia was test positive, while the remaining 2 isolates were test negative, with the exception of Cryptococcus laurent. All isolates showed a positive result in a test to determine the viability of growth in low level of water with increasing sodium chloride levels. Regarding the results of Table 2 shows the ability of all yeast isolates to grow at temperature of 25 and 37, except for Cryptococcus laurent, which was gross negative at 37. The table data also reveals the ability of Rhodotella rubra and Cryptococcus laurent was shown to benefit from nitrate as the only source of nitrogen, as it grew weakly and the remaining two isolates were tested negative. Regarding the determination of the ability to resist acetic acid, all isolates were negative for the test, except for Rhodotella rubra, which was positive for the test. As for the test to determine the viability of growth in low level of water with a rise in the level of carbohydrates, it was found that Rhodotella rubra and Trichosporon achea was test positive, while the remaining two isolates were test negative, with the exception of Cryptococcus laurent. All isolates showed a positive result in a test to determine the viability of growth in low level of water with increased sodium chloride level. Regarding the diazonium blue B DBB color test, all the isolates were negative for the test, and this indicates that they belong to the Ascomycetes, except for Rhodotella rubra, which was positive for the test, indicating that it belonged to the Yeasts of the Basidiomycetes. The results of mutagenesis of the isolates were showing ethyl methyl sulfonate at a concentration of 0.2 mg per ml has a clear effect on the viability of all types of yeasts exposed to it. The highest killing rate was 72% in Trichosporon achea, while the lowest was 47.4% in Cryptococcus laurent. It is evident from the observation of table 4 that Rhodotella rubra was resistant to all antibiotic use except for Lamisil. The rest of the yeasts differed among themselves in their ability to resist antibiotics. It is clear from table 5 that Candida tropicalis became resistant to all antibiotics, while Trichosporon achea became sensitive to all antibiotics except chlorotrimazole, while Rhodotella rubra and Cryptococcus laurent is sensitive to all antibiotics and has retained the antibiotic resistance status of candizole. Thank you for your attention.